Vitamin B12 supports nerve health, red blood cell production, and DNA synthesis. Genetic factors can affect B12 absorption, transport, and utilization within cells. One of the most common genetic predispositions affecting absorption occurs in the fucosyltransferase 2 gene, also known as FUT2. In a meta-analysis involving over 3,600 subjects, the G allele was strongly associated with lower B12 blood levels. Scientists believe that this FUT2 variant reduces B12 absorption by altering the gut microbiome. Once vitamin B12 reaches the bloodstream, carrier proteins deliver the nutrient to cells that need it. One of these proteins is called transcobalamin 2, encoded by the TCN2 gene. A polymorphism in this gene reduces circulating levels by as much as 19%, according to a meta-analysis of 7 studies and over 1,400 individuals. One of the major intracellular functions of B12 is to support the methylation of homocysteine. This process involves 2 enzymes, methionine synthase, encoded by the MTR gene, and methionine synthase reductase, encoded by the MTRR gene. 2 genetic variations affecting these enzymes may potentially increase B12 demands. Further assessment of B12 status is recommended for carriers of any of these variants to confirm the need for nutritional support.